Now let's learn about the confocal microscope.
Is it like this one?
Well, it is a type of light microscope like the ones we use in school, but instead of using a light bulb to see things, it uses lasers.
Ooh, lasers.
Pew, pew, pew.
So how exactly do you see things with lasers?
First, you need to become fluorescent.
You mean we'll glow in the dark?
Not exactly. When studying cells like you and me, scientists label us with specific dyes so they can look at specific structures. There are dyes for almost everything. Some will label our nucleus, others our mitochondria, and some will even label our plasma membrane. After staining us, scientists use the confocal microscope to shine a laser at us. After being illuminated by the laser, our labeled organelles will emit light of a different color. To see how this works, let's take a closer look at my mitochondria. A scientist labeled my mitochondria with a stain called mitotracker red. When a laser illuminates this stain with yellow light, the dye will emit red light. This is called fluorescence. See how this works on me?
It's so pretty. You look so good in red.
One thing about fluorescence is that these dyes almost always absorb light of a shorter wavelength and emit light of a longer wavelength. When you look at a rainbow, red light has the longest wavelength and violet light has the shortest wavelength. With my mitochondria stain, you can see that it absorbs yellow light but emits red light.
Oh, I see. Yellow is the shorter wavelength and red is the longer wavelength.
You got it.
Okay, so let me see if I got this right. With confocal microscopes, you make your sample fluorescent and then use lasers to get an image so you can see organelles and other structures. Does this only work with those stains you mentioned?
Nope, not just with stains. Scientists can genetically alter cells or even organisms, so they produce a fluorescent protein. One of these proteins is called green fluorescent protein, or GFP. It actually originated from a jellyfish.
Wow! What else do confocals do?
Well, confocal microscopes do this awesome thing called optical sectioning.
That sounds dangerous. What's it for?
Optical sectioning is used to get 3D information about a sample.
Oh, cool! How does it work?
When you use the confocal microscope, light from the lasers is focused onto the sample. This light excites the fluorescent molecules in your sample, and the emitted light travels back up through the objective lens, where it eventually hits a component in the microscope called a pinhole. The pinhole is basically a tiny, tiny hole that only lets some light go through. In fact, it only lets through light that is in focus. To get 3D information, use the lasers on the confocal to scan at different depths within the sample. The pinhole only collects the in-focused light at each of those depths, so each image shows a different part of the 3D structure. When you put this stack of images together, you can see the whole sample in 3D.
That sounds awesome. I think this calls for some movies.